This is the video for Lab 14, Multiple Test Media. In building the biochemical profile or enzyme profile for your unknown, up to this point you have used single test media. Single test media means that there is only one substrate in each type of media. This lab is going to utilize multiple test media. So for multiple test media, that means you have more than one substrate in each type of media. Also, there will be more than one indicator in the media. And you will be looking at more than one reaction in the media. The first type of media you would have used is the triple sugar iron agar, or the TSI. And for this triple sugar, you are looking for your bacteria's ability to utilize glucose, sucrose, and lactose. And the indicator for these three sugars is phenol red. So that's very similar to the experiments you did in single test media. So if the bacteria are able to utilize the glucose, sucrose, and or lactose, they will release acidic byproducts and turn the media yellow. The other test you are running is to determine the presence of cysteine desulfurase. And if you remember, you are looking for the production of hydrogen sulfide that indicates that the cysteine desulfurase is breaking down the cysteine. And the indicator that is used is iron. So if the cysteine desulfurase breaks down the cysteine, producing hydrogen sulfide, that hydrogen sulfide will interact with the iron, turning the media black. Interpretation for multiple test media can be very complicated. And in your lab, you have a table like this, which will help you identify and interpret the different results. For this, you can look at these abbreviations. K means alkaline, A means acid, G means gas, and the S means the hydrogen sulfide production. For the triple sugar iron agar test, the original media would have been in a slant in a test tube, and the color would have been red. And to inoculate it, you would have stabbed down to the bottom of the tube, and then as you brought the needle back up, you would have inoculated the slant. There are many different types of results you can see in a TSI tube, and these are some of the results. When going through and evaluating them, you're looking at the color in two parts of the lab tube. One part is the butt. The butt is the bottom of the tube, and then we talk about the slant, which is the rest of the tube. In this first tube here, the butt is yellow, which indicates that glucose was utilized. The slant is yellow, which means that sucrose and or lactose was also utilized. In the next tube, the butt is black, which means hydrogen sulfide was produced. And then the slant is yellow, which means sucrose and or lactose was broken down. The next tube is all red, which indicates negative for glucose because the butt is still red and the slant is still red, so negative for lactose and sucrose. There's no blackening. Then the next one, the butt is yellow, so positive for glucose. The slant is red, so it's negative for lactose and sucrose. In the next one, the butt is yellow, so that's positive for glucose, and you see this air bubble, so that means it's also positive for gas. And the slant is red, so it's negative for lactose and sucrose. Here again, you see a yellow butt and cracks, so it is positive for glucose, positive for gas, and the slant is yellow, so it's positive for sucrose and or lactose. And here, it looks like I think there is gas. So if you have gas, then glucose had to have been utilized. You see the black for a positive hydrogen sulfide, yellow slant, positive for sucrose and or lactose. The second test you would have done would be the sulfur indole motility, or SIM Agar. And for this, you are looking at the results for 3 different tests. The first one is similar to the TSI, and you are looking for the presence of cysteine desulfurase, which again is breaking down the cysteine to release hydrogen sulfide gas. So iron is the indicator that is used. And if it is positive for the cysteine desulfurase, the media will turn black. Also, for the indole test, you are looking for the presence of tryptophanase. And for this, the tryptophanase will perform a reaction and release indole. In order to identify the presence of indole, you need to use Kovacs reagent. And then the last test is motility, so looking for the presence of the flagella. Again, interpretation can be complicated, so in your lab there is this table which breaks down the 3 tests, so sulfur reduction, indole production, and motility. So you can use this information to help you interpret the results. So for the sulfur indole motility assay, the original media would have looked like this, and to inoculate it, you would have stabbed down to the center and then inoculated it that way. After 48 hours of incubation, you would have added some Kovacs reagent to the top to look for the reaction. Some possible results. So sulfur, indole, and motility. For this, the Kovacs reagent turns pink, so that is a positive. There's no blackening, so it's negative for sulfur, and it looks like the bacteria is pretty much non-motile. The next tube, Kovacs reagent remains that yellowish greenish color, so it's negative. There's no blackening, and this is definitely non-motile. In this tube, there is definite blackening, so it's positive for hydrogen sulfide production, and the Kovacs is yellow, so negative for indole. For this one, indole is still negative, the Kovacs is yellow, there's no blackening, so sulfur is negative, and again, this looks non-motile. And in the last one, Kovacs turns pink, so that is positive for indole. There's definite blackening, so it's positive for hydrogen sulfide production. And motility, it looks like the growth is moving away from the stab line. For the SIM media, motility can be difficult to read because we have not added the indicator that turns pink.